Hello everyone, I'm honored to be here and appreciate ICS chairman and my boss and colleagues. I have no COI. Today, I'm talking about Nocturia, especially in nocturnal polyuria. Nocturia impairs quality of life and increases mortality. 50 to 80% of nocturia is caused by nocturnal polyuria. However, clinical treatment for nocturnal polyuria is limited. Pathophysiological mechanism is not well understood, partly due to the lack of optimal preclinical models. We aim to establish a novel nocturnal polyuria animal model and elucidate the molecular mechanism. This is the research process. Due to time constraint, please refer to the abstract for details. First, establish a novel nocturnal polyuria animal model. In recent years, salt intake positively correlates with nocturnal polyuria in the topics. However, although salt intake amount does not vary with age, the number of subjects with nocturnal polyuria increases with age. From this fact, we hypothesized aging has a major impact on the development of nocturnal polyuria as well as salt intake. There are several factors which affect aging, such as SIRT1, chlorosol, nitric oxide, NO, and so on. We focused on NO. NO decline is well known to induce endothelial dysfunction and neurological dysfunction. We examined whether NO production affects the relationship between salt intake and nocturnal polyuria in humans. As we expected, nocturnal polyuria was associated with salt intake only in subjects with low NO levels. We hypothesized combination of NO deficiency and high salt intake induces nocturnal polyuria. We used 90 weeks mice. Mice were given either normal water, LNM, an inhibitor of NO synthase, and either normal salt diet, NSD, or high salt diet, HSD, and divided in 4 groups, and 2 weeks later we analyzed. To measure urine volume, we used the AVSOP method. Mice are kept on a wire mesh for 4 days. A roll of paper under the mesh catches urine, and the amount and time of urine is measured. We defined diurnal polyuria index (DPI), calculated by dividing inactive period urine volume by 24-hour urine volume. Because mice are nocturnal, DPI corresponds to the human nocturnal polyuria index. Under tap water, DPI did not change after salt loading, and 24-hour urine volume did not change. On the other hand, under LNM, DPI increased after salt loading, and 24-hour urine volume did not change. We have successfully established a novel animal model of nocturnal polyuria by combining NO deficiency and high salt diet. Second, elucidate the molecular mechanism. This slide shows you about urinary sodium excretion. In the active period, salt loading increased sodium excretion under both tap water and LNM. Salt loading under LNM suppressed the sodium excretion increase. On the other hand, in the inactive period, salt loading under tap water did not increase sodium excretion. Salt loading under LNM significantly increased sodium excretion. Sodium and water are reabsorbed at various sites. NCC in distal tubule and ENaC in collecting ducts are main regulators of sodium balance in the kidney. We evaluated the transporters by Western blotting. This is the result. Phosphorylated NCC decreased after salt loading and tap water. What we are interested in was phosphorylated NCC does not decrease after throat loading under LNAME. Next, we investigated upstream pathway. SPAK is currently the only non-activator of NCC, and aldosterone-angiotensinogen regulates SPAK-NCC pathway. We evaluated serum aldosterone and renal angiotensinogen. This is the result. Throat loading decreased serum aldosterone levels under both tap water and LNAME. Throat loading did not change angiotensinogen under tap water. Throat loading increased angiotensinogen under LNAME. Lastly, we administered thiazide, an inhibitor of NCC, and confirmed thiazide improved nocturnal polyuria in the animal model. This is a summary of our research. As shown on the left, throat loading in normal condition decreased serum aldosterone and suppressed phosphorylated NCC in the active period and induced sufficient sodium excretion. On the other hand, as shown in the figure on the right, throat loading under NO deficiency increased intrarenal angiotensin II and could not suppress phosphorylated NCC and caused insufficient sodium excretion in the active period. And finally, increased urine volume in the active period. In conclusion, we have successfully established a novel animal model of nocturnal polyuria by combining NO deficiency and a high salt diet. The intrarenal RAS-SPAK-NCC pathway could be a promising therapeutic target for nocturnal polyuria.